Hello everyone. Have you ever wondered how HPLC instrument can separate and identify multiple compounds from a single sample with such incredible precision? But what actually happens inside the HPLC? From the moment the sample is injected to the moment those peaks appear on the chromatogram. In this video, we will break down HPLC chromatography from the basics, its principle, every major instrument component, complete working process, and how the final chromatogram is generated. So if you want to understand what is really happening inside an HPLC, stay with me till the end. Let's decode HPLC step by step. Introduction of HPLC. High performance liquid chromatography, commonly known as HPLC, is one of the most widely used analytical techniques in pharmaceutical quality control. It is used for identification, assay, impurity determination, related substance, dissolution, degradation product analysis, and many other applications. Basic principle of HPLC. The basic principle of HPLC is separation. A sample containing different chemical components is injected into a flowing mobile phase. The mobile phase carries the sample through a stationary phase packed inside the HPLC column. Because different compounds interact differently with the stationary phase and mobile phase, they travel through the column at different rates and are separated. Mobile phase preparation. The first step is preparation of the mobile phase according to the approved analytical method. The mobile phase may contain water, buffer, methanol, acetonitrile, or other suitable solvents in a specified proportion. Filtration and degassing. Filtration and degassing of mobile phase. After preparation, the mobile phase is generally filtered through an appropriate membrane filter to remove particulate matter. It is then degassed to remove dissolved gases. Filtration helps protect the HPLC system and column, while degassing helps minimize problem caused by air bubble during analysis. HPLC system startup. Before starting the analysis, the HPLC system is checked for proper connections, solvent availability, leaks, column leakage, and instrument status. The appropriate mobile phase is placed in the solvent reservoir and the solvent line are properly primed. Pump and flow. The pump delivers the mobile phase through the HPLC system at a controlled flow rate. Depending on the analytical method, the system may operate under isocratic or gradient conditions. Column. The column is the heart of the HPLC separation. It contains the stationary phase which interacts with the components of the sample. Different compounds interact differently with the stationary phase, resulting in different retention times. Standard preparation. A reference standard or working standard is prepared according to the method. The standard concentration must be accurately calculated because the standard response is used for quantitative determination of the sample. Sample preparation. The sample is prepared according to the approved analytical method. Depending on the test, the sample may be accurately weighed, transferred into a volumetric flask, dissolved using the specified diluent, sonicated if required, and diluted to the required concentration. System suitability. Before analyzing samples, system suitability is performed to demonstrate that the HPLC system is capable of producing reliable and reproducible results. Injection. Once system suitability is satisfactory, the sample vial is placed in the autosampler. The autosampler withdraw a defined volume of sample and introduce it into the mobile phase flow. Separation inside the column. As the injected sample travels through the column, its components interact differently with the stationary phase. Some components interact strongly and take longer to elute, while others interact less strongly and elute earlier. Detector. After separation, the individual components enter the detector. The detector measures the response of the compound as they elute from the column. In pharmaceutical HPLC, UV or PDA detectors are commonly used, although other detector types are also available. Chromatogram. The detector response is converted by the data system into a chromatogram. Each peak generally represents a component detected at a particular retention time. Retention time. Retention time is the time taken by a compound to travel through the HPLC system from injection to detection. Under controlled chromatographic condition, retention time can help identify a compound by comparison with an appropriate reference standard or working standard. Peak area and quantification. For quantitative analysis, the peak area is commonly used. The response from the sample is compared with the response from the standard according to the calculation specified in the analytical method. Impurity analysis. HPLC can also be used for related substances or impurity analysis. In this case, a small peak other than the main component may be evaluated according to the analytical method. Impurity identification and quantification depend on the validated method, reference standard, response factors, and applicable specification. Chromatogram review. After completion of the sequence, the chromatographic data are reviewed. The analyst checks system suitability, chromatographic performance, integration, identification, calculations, and compliance with the applicable acceptance criteria. Final result. Finally, the calculated results are compared with the approved specification or acceptance criteria. The result is then documented according to the laboratory data integrity and quality system requirements. Complete HPLC workflow. In summary, HPLC is a complete analytical process involving mobile phase preparation, standard and sample preparation, controlled chromatographic separation, detection, data acquisition, calculation, and result evaluation. Proper control of every step is essential to obtain accurate, precise, reliable, and scientifically sound analytical results. If you found this video helpful, don't forget to like, share, and subscribe.